biology for our spatial platforms, you don't have to remember a bunch of different chemistries. The chemistry's all the same. It's basically all the same chemistry, all the way from our encounter hybridization. You can see there at the bottom of the screen. Everything that we do at NanoString is hybridization, and it all works with these small little areas of 35 to 50 nucleotides. And CosMx chemistry on the right is that we get our hybridization. There's no enzymes, no amplification, just a hybridization. And then we read it out with imaging barcodes. These are special barcodes that we made for imaging. They are basically the equivalent of the encounter barcodes that were 2 microns long. And now we make an imaging barcode that is 20 nanometers big. So that's how far we've condensed it out into the CosMx imager. And then you see the GeoMx hybridization, the same overall hybridization chemistry. In fact, it's the same design, literally, except now we're not imaging the individual transcripts. We have sequencing tags with photocleavable linkers on them. And then we read, it's an in situ hybridization, but we read it ex situ on a next-gen sequencer. But again, just hopefully that it's nice talking to a user group, but you can really sort of understand this, is that the way we do hybridization design, the probes we make are identical as you move through all these different platforms. So what that means is that you could discover some brand new discovery on a CosMx, and then say you wanted to do a large patient cohort on hundreds of clinical samples, and you could do it in GeoMx, and you would not have to change the business end of your molecule. It's identical between CosMx and GeoMx, and do your large clinical cohort study. And then, who knows, you may find something you could actually make a in vitro diagnostic or a laboratory developed test that's not spatial with your discoveries. Those probes could go right into an encounter. And again, you'd never have to change your design. And that's really unique, from discovery, a big cohort on translation, and even making a diagnostic. You would never have to change the design of your probes that you're examining, which is very unique. As far as I know, I don't know of any other company where it works that way. Okay, this is something new, and this is about all I can tell you about the new thing that's happening as part of Bruker Spatial Biology. They're very excited about what we can continue to develop on nCounter. And previously, I simply didn't have enough resources in R&D and money to continue developing along all our platforms. And that's changed with Bruker. So they won't let me tell you anything else other than you're going to see some really exciting new panels, and you got to keep your eyes open, because I'm excited about doing nCounter research again. When I first came from Life Technologies to start R&D at NanoString 12 years ago when I came, this is what I was doing full time, developing all those panels you see in front of you that were really incredibly amazing panels, and they did an amazing amount of great work in the scientific literature. And I'm back to being able to make some of those panels over again. So super excited about that. Okay, so let's talk about our spatial biology platforms. I'm going to take you through a couple of little vignettes. It's important to realize, sometimes you forget it, or you can forget it, is that all of our platforms, every platform that we make at Bruker Spatial Biology, has been peer-reviewed in the literature, and not just any literature. Every one of them, the encounters in Nature Biotechnology in February 2008. You see GeoMx, digital spatial profiling, in Nature Biotech, May of 2020. And then October 2022, CosMx Spatial Molecular Imager, all peer-reviewed at the highest level, Nature Biotechnology. And these aren't application papers. These are the real instrument papers, how the instrument was made, what the chemistry is. And if you actually look at a lot of the other groups in the field, they actually don't have a single fundamental paper from the groups that actually make the instruments or whatever. But we do. every one of our platforms, the platform itself and the chemistry is all peer reviewed at the highest level. So this is sort of unique to us also. So let's talk a little bit about GeoMx. The new thing I want to describe to you about in GeoMx is this new ultra high-plex protein panel that's never been done in the world before. It's 570 plex. So I'm going to go back to the roots of GeoMx. GeoMx started out as at that time was high-plex protein, and that was 50 markers. Now we're doing 570. Just going to remind you, GeoMx is really unique in the sense that it really pays attention to the geometry of tissues, the geometry of cancer, and you do spatial profiling with these little deformable mirrors, these digital micromirror devices, each with 1,000,000 mirrors the size of your thumbnail. And they sculpt, you take a picture of your tissue, and then they sculpt to be the exact faithful representation of the shape of what's in that tissue that allows you to do full-blown profiling. And if you don't remember, this was actually the very first discoveries we made on GeoMx was on a protein panel. This was actually collected in my lab at Seattle before we even went commercial. That volcano plot you're seeing on the right was the very first clinical trial sample that ever was run on a GeoMx. And this was checkpoint inhibition, and it was a melanoma as a skin cancer. And we were following the responders and the non-responders to checkpoint inhibition and melanoma. And these were the protein biomarkers we found. It was high-plex protein we were doing. And MD Anderson came and I showed them that volcano plot you see to your right. And they said, Beecham, you got something wrong. You got two markers, and they literally circled them on the little Zoom call I was on, and they circled CD20 and CD19. They said, Beecham, we know from our mouse studies there really there's not a B cell component to this response and this inhibition that we're giving here. And I said, well, I see them in biomarkers. They were in our high-plex protein panel. I said, maybe we should look at the images. So we looked at the images closely, and what did we find? Those B cells were associated to a unique histological feature in those tumors. And those histological features are called tertiary lymphoid structures. Literally, almost no one was talking about tertiary lymphoid structures when we made that discovery. We made that discovery. It was published first in Nature Medicine. Then we have back-to-back papers in Nature publishing that tertiary lymphoid structures were the most predictive histological feature for predicting response to immunotherapy. That was our discovery number one. Proteins and GeoMx made that discovery even before full commercial launch. This was our work with MD Anderson. So I've always wanted to take proteins up to the next level. And, I felt okay with things like 50-plex and 100-plex protein, but that's not what I really wanted to do. What I really wanted to do was something like this. We worked closely together with Abcam, which is an antibody company, and we figured out, we developed inside NanoString, we developed whole new ways of barcoding antibodies and high throughput on very low quantities of antibodies, all with a hands-off automated methodology. And that paired up really with Abcam because they had a lot of content. We were able to bring those two things together and make a commercially viable protein panel, antibody panel that has a lot of the features of a high-plex RNA panel. So now instead of just protein with individual biomarkers, we're now talking about proteins that represent entire pathways of biology that we have at 570 plex. So, download the content, take a look at it. We cover all these pathways, and I think it's super exciting to actually see both pharmaceutical companies and a lot of academic researchers very quickly discover new protein-based biomarkers. And you're going to see those papers. I can see them being written now, so they will be out shortly. This is the slide highlighting our teamwork with Abcam. Abcam was very good at doing low-plex, 1-plex very good verification and validation. That's what they did. And I married that pipeline with our High-plex automated verification and validation that works over hundreds of antibodies at a time. And we brought those two things together to make a product. And the nice thing is, it doesn't matter what area of biology you work in, whether normal tissues, cancerous tissues, organ transplantation, autoimmune disease, whatever, no matter what organ you're looking at, and you look at how many proteins I'm observing over all these different tissues, you're going to see on average about 300 proteins above 5x in signal and noise, no matter where you look. So this is really great signal and noise that comes out of this protein panel on a bunch of different cases. And then when we're making this high-plex protein panel that's never been done before at this plex, we wanted to be compatible with the same slide 18,000 plex RNA. And we were able to accomplish that. And I got some of the validation data up here. I won't bore you with all of that. But the idea of doing 570 plex protein, 18,000 plex RNA, all on the same slide is really unprecedented. There's no spatial platform doing that at this current time. It's got over 64 phosphospecific antibodies that have also been validated to work on FFP tissue, and I'm super excited about the results that can come from this. Yeah, I was given a talk the other day, and this was a data that was being shown to us from Arutha Kulasinga from Queensland University in Australia. And this is what head and neck cancer looks like at 570 plex protein. Just look at the top. Look at the dynamic range of all the protein markers that are being examined, all fitting into these unique areas of how head and neck cancer responds. All the principal component analysis and things you can do where you can look at normal and disease states, all give you beautiful separation purely on the high-plex protein information. And then you can make responders and non-responders graphs out of pure proteins that look like volcano plants, graphs that typically come out of RNA work, now showing this kind of high density at the protein level. So super excited about that. This one's gotten some airtime, so I won't describe this one in detail, but to give you an idea how robust this is, we had lungs of soldiers that died in over 100 years ago. It was 1918 soldiers, museum quality lung tissue that we could run the IPA together with the whole transcriptome and get high quality data out of those samples. So over 100-year-old FFP tissue, museum quality, we could still get that high-plex protein assay to run and work. And this work's being written up. So I think exciting things that are happening with GeoMx, and in some respects, going back to our roots on what can high-plex protein tell us? And there's still a lot of room for growth there. Okay, so let's talk about CosMx. This is the single cell and subcellular resolution instrument. And there's actually kind of a lot of confusion in this area, so you have to bear with me. But it shouldn't be confusing to anyone. From the very beginning, we designed CosMx to recapitulate all the capabilities of single-cell RNA-seq, but with subcellular resolution. So you don't have to learn a whole bunch of new formulas for what sensitivity is, specificity, and all that. You just use the same things that we've been using for almost a decade in single-cell RNA-seq. Those are still the right stats that you need to know. For sensitivity, you just got to remember one thing. How many transcripts do you get per cell? Okay. And what is the diversity of the transcripts you get per cell? That's the only spec you really got to remember. And that's the one you should ask for. And then you have one other thing, because single cell RNA-seq didn't have to put the RNAs in a single cell because it was all dissociated single cells. Now we need segmentation to put the RNAs in the right place. And I'll show you we're now on our 4th generation segmenter, and there's really nobody that competes with us on the level of segmentation we do. Okay, so let's talk about why plex matters. And this was really part of my design thinking from the beginning. The other competitors for CosMx, when they're coming out, they came out with these organ-specific, essentially custom low-plex panels. This is an example one of 248 plex Xenium panel here. And look at what's white and green there is this is a Voronoi graph of all the tissue biology that can happen, all the reactions associated with tissue biology. And everywhere is white are things you're not measuring. And I'm sitting here doing high-plex spatial biology, and I know that we don't know enough. We just don't know enough information to have all those white zones where I'm not measuring it. So I only became happy when I got to 1,000-plex. I could make a general purpose panel that had enough green in the reactome that I felt that I could make a panel that's not specific for one particular organ or one particular disease. I wanted a general panel that would work over many different types, and that's what we were able to accomplish. We now have over 600 million single cells on this panel. This is a little bit dated, and growing, and literally 100 publications that come out in a wide variety of places. You can see inflammation, you can see organ transplantation, you can read cell atlasing, and you can do it all with this general purpose 1,000-plex panel that we came up with. And I think that was really the right decision that we made, because now you can compare your data sets. If you are an autoimmune disease person, did CosMx 1,000-plex, you can have a conversation with a cancer researcher that used that 1,000-plex, same one, to study an ovarian cancer or whatever. And you can look at immune signals that are common between these two different types of diseases. Simply not possible with low-plex custom panels to really compare. And that's really where you learn so much. I grabbed just one of the recent ones that came out. This was a fundamental discovery made with the 1,000-plex panel. You can see here another Nature paper where we discovered a unique aspect of the macrophage inflammation and how how it is regulated and what information is going on there. Again, another Nature paper from one of our 1,000-plex panels, and that just came out recently. So let's talk a little bit about the comparison studies. There was a lot of noise made about a couple comparison studies, and they were saying just really they're making some claims on comparing platforms that have pretty much later been shown to be intrinsically flawed from the beginning. And I'll show you the one study you can see on the left-hand side was done with one FFP tissue. And from an N=1 study, you cannot make a platform to platform comparison. So that's intrinsically flawed because it's an N=1 study. And then the one on the right is intrinsically flawed because it turned out they're comparing, you can see Xenium, Merscope, and CosMx all being compared. But the time between when they cut the section and when it went into measurements that they wrote up in this paper were all different. CosMx sat around for 44 days before they made any measurements on them. Poor Merscope had a case where they went 74 days before it was measured. See, they were writing this paper as if you could systematically compare the platforms. In reality, what they were doing was basically just measuring the effect of cutting and waiting 44 days. So these were intrinsically flawed comparison studies. Luckily, some scientists out there started doing good comparison studies. And these were one of the very first ones that came out. Parambir Delai, who's an MD at Northwestern University, said, I'm going to do it right. It's got 16 blocks, 11 different patients, samples. They each get cut at 5 microns. The day they're cut, one of them goes into the Xenium, one of them goes into the CosMx. And then the data are compared and they looked at head to head. So now we got some statistics and we can actually see what's happening. So what did you observe? Yeah, they observed that CosMx had about 2.5 times the sensitivity in terms of net transcripts observed, 4 times the gene diversity of genomic breadth of the samples. And you can see, and again, these are averaged over 16 samples, 11 different patients. So there's some real statistics behind that. It fit up very well with an N=1, similar N=1 study, CosMx Xenium, that was done actually in Japan by Yataka Suzuki. But you can see it's very much. So now we got the statistics on something now where you can really, you can really look at your errors and what you observed. Pear and Beer looked closely at the overlapping genes, and then they found that ours actually were more sensitive. CosMx was more sensitive than Xenium on the overlapping genes, but I consider it's kind of close. But when you look on the right-hand side, I take any data analyst you want. That's the violin plot, CosMx in green, Xenium in blue, on transcripts per cell on the left, and then the unique genes per cell. And show that to your data analytics person and say, which data set do you want? Do you want the green violin plots or you want the blue ones to try to understand what's happening in your tissue? I guarantee you there's not a single data analyst that's going to vote for the blue violin plots. They're all going to take the information content in green. Okay, that group also saw the segmentation errors. They used our 4th generation segmenter so that the transcripts are going into the right cells. And, anyway, a nice comparison study. Now that one's still not out and published. So luckily we had another one that is out and published now that I can actually share with you. This is a recent paper that came out in Nature Immunology. And then look at the unique way it was designed. CosMx was the discovery. CosMx SMI was the discovery. They're doing 1,000-plex. They profiled over 100 tissues with that. That was their discovery platform. And what they did is they went back and they validated with low-plex Xenium and relatively low-plex MERFISH. So what this is a great way of saying, okay, well then what did they observe? And you can go in there and examine this data yourself. First, they made the discovery. The key discovery they made is in the figure G there that you're looking at. That is learning all the ligand receptor conversations that are happening between activated CD8 T cells and all those pathways around it. Now, Xenium and MERFISH didn't have enough content in there to do that kind of discovery, but they were able to validate one gene at a time on the overlapping genes, and I'll show you what that looks like. So again, these are data sets that are in the public domain. You can go ahead and look at them yourselves. You get transcripts per cell, CosMx and Xenium, unique genes, and I'll show you what the overlapping plots are so it's easier to see. Again, these are the transcripts per cell, CosMx in orange, Xenium down here in blue, and then the unique genes per cell, CosMx ahead, Xenium down there in blue. And now you can see all these overlapping on the overlapping genes, and it turns out there's 113 overlapping genes, all these different patients, 2 different areas, topological regions, and on the overlapping genes, yeah, they're pretty much commensurate. Sometimes CosMx a little higher, sometimes Xenium is, but it's pretty much commensurate among this whole batch. So don't worry about those. Those previous papers that were flawed with all their sensitivity differences, it doesn't get held up when you really do the study correctly. And you could actually go, this is a public domain data set. They set up this data inside a Seurat object, so you can go ahead and make those exact same graphs. So you don't have to take my word for all these comparisons. You can actually see it for yourself. These are all the correlation plots on the individual genes, on the overlapping cases. Everywhere they found that they validated each other. So there was no crazy false discovery rate happening out of CosMx. Didn't exist because that's not really that's not a discovery rate statistics for spatial. So everything validated and it worked fine. And again, this is CosMx essentially high-plex, still getting compared to Xenium low-plex. Okay. And finally, Xenium higher-plex data has come out. all the way up to 5,000 plex. So let's look at what those comparisons look like. Now I don't have to correct for these big plex differences. So what's it look like? Now, it turned out that the way CosMx was designed, it's got the same protocol no matter what plex you do. It's exactly the same. It was designed so our 1,000 plex, 6,000 plex, and whole transcriptome imaging CosMx has exactly the same workflow. You hybridize, you light up your protein segmentation markers, and you read it out. That's it. Simple. Now, when Xenium went high-plex with the rolling circle amplification, they actually had to add a whole new series of steps that didn't exist before. Those are labeled with the little stars, the little red stars. They got an extra primer hybridization step, another RNA step, a step called polishing, a brand new that just takes into account they're trying to get to high-plex with this technology. And then those little forward-looking yellow arrows means this is all stuff you're doing off the instrument with your own hands. Those are steps that have to be done really timing critical with each other. So there's all these extra steps and extra timing specific steps that are added to accomplish the high-plex Xenium 5,000 plex panel. So let's look at how they compare. We got more content, so we get 23% more diversity of transcripts. Okay, how do you expect that? But also, we've been designing panels like this all the way back from Encounter days. So we're really good at capturing biological information in our panel. So our 6,000 plex actually has 84% more information associated with ligand receptor pair signaling than the Xenium 5K. When you look at the reactome coverage, which is pathway biology, you actually got 81% more pathway biology in our panel. So intrinsically from the design, you get an advantage. And then let's look at what the transcript comparisons are. Again, this is Parambir Delay doing it exactly the way he did that very good comparison on 1,000 plex versus the Xenium. He's now got 6K and 5K data done exactly the same way. Tissues cut, shared in two places, and then what do you get? And here's where you get sensitivity ratio of CosMx to Xenium. Now we're both at nearly the same plex, so this is pure sensitivity increase. We're about 3.5 times more sensitive in transcripts per cell, 819 transcripts per single cell. Xenium averaged 235 on that. Unique genes, the diversity, we got about 3 times the breadth of unique genes. And let's look at the overlapping. The overlapping genes now is 2,550 comparisons of the overlapping. And what do you see? Now there's an increase in intrinsic sensitivity of the CosMx over Xenium, over twice as sensitive on the overlapping genes now as the Xenium. So it used to be commensurate when Xenium was low plex, but as they go to high plex, the CosMx sensitivity continues to increase because we made that chemistry for high plex technology. It was born to do high plex. And you can see the sensitivity start to diverge. We become even more sensitive on the overlapping genes as you go to higher. And you can say, why? Why is that the case? And this is really, you have to get back to how the chemistry works. What really matters on an imager? What are you doing? You're imaging in the spot associated with individual transcripts is what you're measuring. So you got to say that where do the spots come from? Okay. In CosMx, the individual spots are actually made in a factory. So all the imaging barcodes are made with exactly 30 fluorescent dyes per barcode. And actually the imaging barcodes are size exclusion purified. They're exactly 20 nanometers in size. They don't change their shape or their intensity. Now, if you're looking at a rolling circle amplification, those are those things that are actually happening to the tissue. You can think of it as that you're making the detection reagents on the tissue one at a time, essentially, the technician running. So the size of the individual transcripts change because as you amplify, the amplification circles are different depending on what transcripts you're amplifying and where in the tissue they are at. So you get that variability. And as the size changes, the intensity changes. And this was noted in the literature, so you don't have to take my word for it. If you look at the paper to the right, you can read there, due to the complex environment of crowded proteins and other biomolecules in cells, the amplification efficiency of rolling circle amplification is not uniform. And then they show the diversity intensity of a typical RCA that you're doing on a cell in space. And the intensity fluctuations is about 3x. That means your intensity of your individual spots are going to vary by 3 times, depending on the efficiency of rolling circle amplification on the tissue. Of course, we don't have any of that. All the intensities in CosMx are identical, 30 dyes, not 29. It's not 31, it's 30. All the sizes of the spots are identical. I make them in a manufacturing plant here in Seattle. They're all 20 nanometers. They're all the same size. And I think that's some of the origins of why our the tissue signals for us at High-plex continue to go up, where in Xenium it doesn't go up nearly as much. In fact, if you look at the Xenium High-plex 5000 plex and FFP tissue, it actually compares to about the lower third of a CosMx 1K experiment on FFP tissue. Okay, so 5K Xenium is like the lower third of a CosMx 1K panel on FFP tissue. That's the difference. Okay, so I'm gonna have to go fast here. Segmentation has always been super important to us. We're working on our 4th generation segmenter, whereas, yeah, I mean, it's the 1st generation segmenter that's ever been built for the Xenium is just happening now. That's a real data-driven multi-omic segmenter. But like I said, I'm on the 4th generation of that software, and you can see things like this. You'll not see anybody segment cells like this. Let me just show you a couple of pictures in the brain. You can see that. I like this next one. Look at those astrocytes. And look at the protein-driven segmentation of that plasma membrane. You're really following the unique geometry of these cells now. And so you can look at the RNA that's way out on a protrusion from an astrocyte and say, how's that compare what's in the cell body? Some of these cell bodies in the brain can be as long as 3, 4, 5 millimeters projection from a cell body. And literally no one now, everybody's just quantitating. And when you're in the brain, they're just quantitating in the cell body. No one is actually looking at the transcripts far out and these large protrusions, but that's what we do. That's where our segmenter works, and you can pick up all these transcripts that are way out in these protrusion regions. So very unique, and we continue to advance the segmenters. And I'm going to briefly discuss 6000 plex. So what are some of the initial discoveries being made with 6000 plex, just to give you an idea on the kind of discoveries that are being made now at 6K. I think the beauty when you're imaging at this plex, it's important to think differently. You're not thinking about individual genes and marker genes. What you should be thinking about is projecting entire biological pathways onto your tissue. So you can look at that one right in the middle. All I'm doing is taking the reactome for glucose metabolism, projecting it down onto the tissue here. This is a colorectal sample I'm looking at. And look at all the light green areas. Okay, what do those things represent? What those represent, every tumor cell in that colorectal is being lit up by its own intrinsic differences in glucose metabolism. Probably all the cancer researchers remember something called the Warburg effect. There's an imaging of the Warburg effect in space. Now, the beauty with all those pathways, there's all sorts of undiscovered pathways that map like this in tissue, but no one's ever examined them, but now you can. You can project PD-1 signaling, cell cycles, stemness. I could see the muscles with my eye on that. You can see the muscularis propria in the colorectal one. And I said, well, let's just project muscle contraction onto that. Maybe I wasn't I hadn't looked at enough pathology segments to know where muscle is versus where it wasn't. And you can see muscle contraction maps directly to the muscularis propria there. So I didn't need to know that. I didn't have to be a pathologist that can recognize muscle. The pathways themselves will tell you where things are at. So very unique. You just have to think a little bit differently. So as per usual, we always go to a hardest case. Pancreatic cancer is one of the most difficult cases, samples to look at. So we put 6K on it, and this is our first discovery we made there. And I think it's fundamental. You know, everybody think about cancer panels that you have heard about. Does anybody put nerve development or nerve development genes in their cancer panels? The answer is no, nobody did. That doesn't mean they're not in there doing something important. So at 6,000 plex, I got the area of the Verneuil graph where there's actually over 2,000 nerve development genes in our 6,000 plex panel because we're covering all the biology. And what do we discover? Nerve cells actively talking to both the tumor and the immune cells in the stromal regions of pancreatic cancer tissue. Totally unexpected. No one even thought it was going to be in there, but that's the value of having a high-plex. We made that discovery. Now, when we made that discovery, you got all these pancreatic cancer experts coming out to us and say, "Oh, I understand. I can understand why my patients have this kind of pain, and that pain is a direct reflection of whether they're going to survive or not. Actually, and now they got a molecular reason for it because the nerves are in there." In the stromal region, they're signaling both directions, and now they got a molecular answer for something that they used to just notice in their patients. Some pancreatic patients have very painful tumors. Okay, this is one other one that discovery we made. This is with MD Anderson. We were looking at metastasis from esophageal cancers into the brain. And we're imaging at 6,000 plex, but one of the beauties of CosMx spatial molecular imager is that we can do subcellular imaging. We made spike-in probes that were made for bacteria. Okay, so look over on that little right-hand side rotating. What I'm looking at in that cell there, we counted 49 individual bacteria on the intracellular on a single on a cell on this brain met in this particular patient case, and it wasn't the only one. There's a lot of controversy in this field about the microbiome affecting cancer. Are they there? Are they not? This unequivocally shows there's intracellular bacteria in some of these tumor cells. We mapped them over many different tumor cases. They predominantly map with the tumor cells, but we can make three-dimensional maps of the intracellular bacteria with CosMx, and you get these absolutely gorgeous three-dimensional images. So sort of excited about that discovery that we made also. Okay, so now let's talk about what kind of discoveries we're making at the whole transcriptome level. You know, the beauty of doing this whole transcriptome is I've already done this in GeoMx. So all the model systems I had, all the ways of measuring sensitivity and specificity, I had those same materials. I could do the same quantitation over RNA-seq of these unique cell lines that we've been examining for years. So you can just see how we developed it and debugged it. One thing that was really interesting is look at that item number 3 there. Look at that cell over on the right there. That's a fixed cell in culture. That's a LinCap cell that we're looking at here, sort of a model prostate cancer cell line that a lot of research gets done on. I'm counting over 10,000 transcripts in that one single cell. 10,000 transcripts. And that's from the whole transcriptome. Yeah, that's really incredible high resolution data that's coming out. So we made this data public, so I won't talk too much about it. But I love pancreas because you have these regions of acinar cells where you shouldn't see any sugar metabolism. Then you hit these unique features called islets of Langerhans, and you should see all this sugar processing biology come out at you, and that's exactly what you see. So whole transcriptome is working as we designed it. It also allowed us to see some incredibly rare cells that are almost never found in these pancreas called epsilon cells. They're there at 0.008% of the cells, and we were able to quantitate them very uniquely. Okay. And then you actually can say, what are all the private lives conversations that are happening in that pancreas? And we've now done the pancreas at 1,000 plex, 6,000 plex, and whole transcriptome. And the number of above signal ligand receptor conversations at 1K, we could quantitate 404 of them. At 6K, 1,187. At whole transcriptome, we are quantitating 1,486 ligand receptor conversations happening between, as an example, the different cell types you see over on the left-hand side. This is brand new biology. You have no idea all the information that's going to come out of that. And then I like this one a lot. This is early colorectal cancer called tubulovillous adenomas. And we have whole transcriptome images with serial sections of single nuke RNA-seq back to back. And the beauty is, look at the distribution on the right-hand side, the distributions of the transcripts per single cell and the features, which are the diversity of transcripts per single cell. And you can see how the shape distributions line up between the CosMx whole transcriptome spatial image and the statistics for an enzyme amplified single cell RNA-seq experiment. So very happy with that. We measured over 1.4 billion cells at over 1,000 transcripts per cell. And we have literally every one of those cells there, those beautiful green cells in the middle. I got a whole transcriptome on every one of those, the red cells, the orange cells. I've gotten 1.4 million whole transcriptomes on that slide. Just an incredible amount of information. And you can do this also on cells in culture, mixed cells on culture, organoids, 3D cultures, CRISPR-Cas9 engineered cells, and everywhere you look, just the amazing amount of information. And what this really showed to me, like the first comments we got back from Holger Hein when he saw this data is like, why would I ever do a classic single cell RNA-seq experiment again? And there really is almost no reason. You are operating at the whole transcriptome level. You're having up to 10,000 transcripts per cell. You can see there in the middle with all the gene diversity. But now you're doing over 1 million single cells at a time, and you're doing it for less than 1 cent per cell per whole transcriptome. Okay, so not only does it allow you to do easily 1 million cells, you're doing it for less cost, less than 1 cent per cell per whole transcriptome. This is all brand new data that gets enabled by the CosMx whole transcriptome that will be launched early in 2025. So that's not that far away. Okay. So let me just tell you, I'm going to end with just what it feels like to be part of Bruker spatial biology, because additional technologies are going to be coming in to what we can offer. So I talked to you a lot about GeoMx, CosMx, and nCounter, really great at RNA and protein, highest plex on those two analytes. But if you ask me, what were we doing with the DNA in the nucleus, I'd have to say, well, we're not doing anything DNA in the nucleus. Well, part of Bruker spatial biology is a technology called 3D genomic analysis from technology that is part of us in Bruker spatial biology that was called Acuity. So what is Acuity doing? The beauty of what Acuity does is they also believe in the value of hybridization-based approaches. So no enzymes, it's optical barcode hybridization, but all focusing on the nucleus, on the DNA. So you're doing things like you see listed in that. We're literally mapping out the spatial dimensions of all the chromosomes in the nucleus. So we get copy number variations, the gene position on the chromosome, what parts of chromosomes are coming together, what parts are in open structures. All that information is going to come out of the nucleus with the same hybridization approach that we're using in CosMx and GeoMx. So this is a great, now all of a sudden the nucleus isn't invisible to us anymore. We'll be looking in the nucleus. We'll be looking in the cytoplasm, mRNAs, protein PTMs, and putting all that information together. So look for that in this upcoming new year too, where all three of these things will actually be put together. So super excited about the Acuity technology and more hybridization barcodes, but now focusing on the information in a DNA in the nuclear region. And then this is another part of Bruker spatial biology I'm also super excited about is there's an instrument called CellScape. It's a lower plex protein-based instrument, but it really is built from the ground up to sort of have the clinical translation in mind, so that I think the CellScape is going to be a really great way where we can take these discoveries we make in CosMx and GeoMx and nCounter or whatever, and we can actually turn those into high resolution clinical style assays using the CellScape platform. Again, all inside Bruker spatial biology. These are all platforms that all work together. So here is sort of a summary of the GeoMx, Atomics, CosMx part of the thing. I hope I convinced you. GeoMx, highest throughput whole transcriptome, highest plex multi-omic technology that's available. Nothing's really competing with us on that at 570 plex protein. And it's part of the roadmap. We're not done. We're not done with that plexity run in GeoMx. Look for it to continue to go up in 2025. And then CosMx, you can see a lot of the confusion, CosMx, the xenium thing. I hope I put that to rest with a lot of the peer-reviewed work. CosMx has got superior sensitivity on all scales, and that sensitivity is even higher as you go up in plex. We can scale CosMx all the way up to whole transcriptome, and that's launched in the first part of 2025. And then you can see we're incorporating Bruker spatial biology, the Acuity DNA mapping technology, the Canopy CellScape, and the CellScapes running in CLIA labs actually sort of add to the nuclear information and also to the clinical side of our of what we can do as part of Bruker spatial biology. So yeah, I'll leave this one up. Maybe I'll take some questions and answers, but hopefully this is a lot of information, but I wanted to convey a lot of information. There's a lot of new things going on, and all I can say is super good luck with you on your spatial biology research, and